going to discuss about the enzymes and their classification. Yesterday, it was started, and this classification is of two types: old pattern of classification and modern. So, in this nomenclature, an old pattern classification nomenclature was of three types. Adding suffix "ase" to the root word is the first type, and other two types are discussed in our previous class. Then, today, modern method of nomenclature, which is also called as systematic naming. And this systematic naming, systematic nomenclature, it is proposed by International Union of Biochemistry. And this, according to this, each and every enzyme name that can be prepared by two parts. And the first part indicates name of the substrate, and the second part that indicates the action or type of reaction. And at the end of this second word, "ase" is added. So, according to the IUB, this classification, nomenclature, IUB nomenclature gives a name to each and every enzyme with two words, in which the first word indicates a substrate name and the second word indicates a type of reaction, and that word is added with the "ase" at its end. So for this, we can take one example, and with the support of one example, we can explain this. And for that, we are taking lactase dehydrogenase is an enzyme. In this, two words are there. Lactase is one word, first word, and dehydrogenase is the second word. This lactase, it indicates the substrate, and dehydrogenase is the second word, which indicates here D means removal. Removal of what? Hydrogenase. That means H2O. That means this enzyme helps in removing one proton or one H2O molecule from the substrate, or to transfer one H2O or proton from the substrate. That's why this enzyme is named as lactase dehydrogenase. So, in this dehydrogenase, the last ASE suffix is added to the second word, end of the second word, ASE is added. So, lactic dehydrogenase converts, and it acts on which substrate? Lactate. This enzyme that converts on the substrate lactate, and that by removing one H plus or H2O, it gives pyruvate. That means transfer of proton from lactate results in the formation of pyruvate. So for this enzyme, required is lactase dehydrogenase. So according to this IUB nomenclature, each and every enzyme has two words or two parts name, in which the first part indicates the substrate name, and the second part that indicates the action of the reaction or type of the reaction, which ends with "ase" suffix. Okay, then with this nomenclature is over. The next, classification of enzymes. According to IUB only, in 1961, this classification was made. And according to this IUB classification, enzymes are classified into 6 groups or 6 classes based on their nature of reaction. That means on which type of reactions they show their effect. And by that nature, these enzymes are classified into 6 types. First one is oxido reductases. First is oxido reductases. The second is transferases. And third is hydrolases. And the fourth is liases. Fifth, isomerases. And sixth, ligases. These are the main important 6 classes of enzymes. And we'll discuss about this each and every one with the examples. First one. Here, the first type of enzymes according to IOB classification made in 1961, oxido reductases. Oxido reductases. Here, this enzymes helps in removing or adding. Adding of what? Means protons or oxygen or electrons from the substrate. That means to shift hydrogen protons or oxygen atoms or electrons from a substrate to the another substrate. For that sake, these enzymes are useful. So, such type of enzymes are called as oxido reductases. So, in this type of oxido reductase type of enzymes, we can take the example cytochrome oxidase as the example. This cytochrome oxidase is an example, and one more example is alcohol dehydrogenase. This alcohol dehydrogenase, we know the meaning of a dehydrogenase. That means D means removal. Always it is removal only. So dehydrogenase, removal of protons, or here protons. So in the presence of alcohol dehydrogenase, alcohol by losing by H2O molecule, it will be converted as acetaldehyde. And here that H2O molecule will be accepted by NAD and turns into NADH2. So here hydrogen proton or H2O is removed from the substrate. And this alcohol, in this example, alcohol we are taking as substrate. So alcohol converts into aldehyde by losing one H2O molecule, and that H2O, from that H2O molecule, H2 or protons are accepted by NAD and that changes into NADH2. So here this enzyme is acting as oxidoreductases or reductase enzyme. So oxidoreductases are the enzymes which helps in removing or adding of protons or oxygen or electrons from one substrate to the another substrate. So based on this type of nature of this enzyme, these are grouped into oxidoreductases. This enzyme is helping in oxidation or reduction reactions. So type of reaction is oxidation or reduction, and this enzyme is participating in this type of reaction. That's why they are called as oxidoreductase enzymes. Then second type of enzyme, according to IUB classification, the second type of enzyme is transferase. Here itself, this heading itself is indicating what is that? It helps in transferring. And transfers what? It transfers what? Means enzyme that helps in transferring functional groups like phosphate groups or sulfur groups, etc. So here in oxidoreductases, they remove the water molecule, but here transfer, that means they transfers the functional groups from one substrate to the another substrate. So transfer of functional groups, example phosphate groups or sulfur groups are another type of example. So amino groups, etc. So that functional groups are shifted from one substrate to the another substrate. That's why these enzymes are called as transferases. And here, example is taken as glucose. Glucose in the presence of hexokinase enzyme and utilizing one ATP molecule. That means adenosine from adenosine triphosphate, it takes one inorganic phosphate radical, and by combining with the inorganic phosphate radical, this glucose turns into glucose 6 phosphate. Here, phosphate group is functional group of ATP. Inorganic phosphate or phosphate group is removed from the ATP and it is added to the substrate. So here glucose, by taking one inorganic phosphate, it changes into glucose 6 phosphate. So transfer of functional group. Here transfer of functional group from one substrate to the another substrate. And this ATP, by losing one inorganic phosphate, it becomes as ADP, adenosine diphosphate. Here triphosphate, 3 phosphate groups are attached with this, but by losing one IP, it becomes ADP. So phosphate group is shifted from one place to the another place. For this enzyme, which helps, is called as transferase. Here the enzyme is hexokinase. And kinase, transfer of phosphate group, and transaminase, transfer of amino group. That means here we have given some examples. Phosphate groups can be shifted from one substrate to the another substrate. Sulfur group also be shifted from one substrate to the another substrate. And amino groups also be shifted from one substrate to the another substrate. And the third one is hydrolases. Third category or third class of IUB classification enzymes are hydrolases. Here, and this hydrolases, and the catalyzes hydrolysis reactions. Catalyzes enzymes helps in hydrolysis reactions, and that is a breakdown of covalent bonds by introduction of water. That means by adding one H2O molecule, they breaks the bonds between the molecules. Here, example, this digestive enzyme like amylase, cellulose, and sucrose, all these are comes under hydrolases. And for example, if you take sucrose, by adding with one water molecule in the presence of enzyme sucrase, they turns into changes into monosaccharides like glucose and fructose. That means for splitting of bonds, this H2O molecule is utilized. So hydrolysis means by utilizing one H2O molecule, this bonds between the substrate molecules will be separated, and that is called hydrolysis. Through hydrolysis reactions, and this enzyme catalyzes the reactions, and that splits the complex molecules into simple molecules by breaking the bonds with the support of H2O molecule. Such type of enzymes are called as hydrolases. Best example, so sucrose, which is a complex molecule, sugar molecule, and with the support of H2O, it splits into monosaccharides like glucose and fructose. Then fourth type of enzymes, lyases. This catalyzes non-hydrolytic removal of functional group from substrate to the cleavage of covalent bond, or reverse reactions, that is addition of functional group across a double bond. See here, this indicates the reversible reactions. That means in forwarded or backward, both are possible in this type of identification. So, catalyzes non-hydrolytic removal of functional group from substrate and a cleavage of covalent bond, or a reverse reaction, that is addition of functional group across a double bond. For example, for this are decarboxylases and aldolases are taken as an example for this lyases. For this example is taken as pyruvate. Pyruvate is a substrate, and here the enzyme is pyruvate decarboxylase. Pyruvate decarboxylase acts as a lyases in a removal group from the substrate. That means from this pyruvate, this enzyme removes carbon dioxide and changes into acetaldehyde. And it is reversible also. That means by adding this carbon dioxide to this acetaldehyde, again it changes into pyruvate. That means this lyases they helps in bidirectional activity. They shows a bidirectional effect. That means that they can forward the reaction or backward the reaction. That means substrate can be changed into end products by removing a functional group, and by adding this functional group, they gives the substrate which participates in the starting of the reaction. So pyruvate, in the presence of pyruvate decarboxylase, it splits into acetaldehyde by losing one carbon dioxide. So that is about the lyases. Lyases especially, they removes functional group from the substrate molecule by a non-hydrolytic process. That means without the participation of H2O molecule, they catalyzes the reaction and the splits the covalent bonds between the molecules, and this is forward reaction. And in backward reaction, again, they adds the functional group across this double bond. So in bidirectional, they are helpful. The next type of enzymes, according to IUB classification, that is isomerases. This isomerases catalyzes a rearrangement of molecular structure to form isomers. Includes epimerization and cis-trans isomerization. That means they converts one isomer to the another isomer. For this, best example is in respiration glycolysis process. In aerobic respiration, the first step is glycolysis. And during this glycolysis process, glucose 6-phosphate, there's a glucose 6-phosphate that changes into fructose 6-phosphate. That means from one isomer to the another, one form of isomer is changed into another form of isomer. That means one isomer is changed into another isomer. Glucose, these are the same, that means both are sugars only, but structurally they are different. That's why this glucose 6-phosphate that is changed into fructose 6-phosphate in the presence of one isomerase enzyme, that is phosphoglucoisomerase enzyme. See here, isomerase enzyme, which helps in shifting one form of isomer into another form of isomer. That's why these enzymes are called as isomers. That means a rearrangement of molecular structure to form isomers. That means they changes the molecular structure, or they rearranges the molecular structure and gives a isomer. And this isomers, they changes from one form to the another form, from one isomer to the another isomer. For this change, enzymes, those who helps in these reactions are called as isomerases. And here in this reaction, that means changing of glucose 6-phosphate into fructose 6-phosphate, enzyme required is phosphoglucoisomerase. So these are another type of molecules, another type of enzymes. The last class of classification of enzymes is ligases. And this enzymes catalyzes synthesis of various bonds coupled with breakdown of energy containing substrates. Usually, ATP adds molecule substrate and means this helps in the formation of new bonds. That means for the creation of new bonds, these enzymes are helpful. For example, pyruvate with the carbon dioxide and utilizing one ATP, adenosine triphosphate, in the presence of enzyme pyruvate carboxylase, it changes into oxaloacetate plus ADP. Why? Because from ATP, one ATP molecule is utilized by the pyruvic acid or pyruvate, and plus ADP plus IP. That means adenosine diphosphate is the resultant of this ATP, and one IP is came out, and this pyruvate turns into oxaloacetate in the presence of pyruvate decarboxylase. Here, pyruvate carboxylase. Carboxylation means by utilization of one CO2. That means one CO2 is added with the pyruvate and that turns into oxaloacetate. And here, energy that splits into ADP plus IP. So, here, new bond is created. That is, from pyruvate, oxaloacetate is produced. So, catalysis, synthesis of various bonds coupled with breakdown of energy. That means ATP that is splitted into ADP plus IP. Containing substrate. Usually ATP adds molecules to substrate. ATP adds molecules to substrate. Means this helps in the formation of new bonds. So overall, ligases are what? Ligases are the enzymes which helps in forming new bonds between the substrate molecules, are called as ligases, and best example is pyruvate plus CO2 plus ATP. In the presence of pyruvate carboxylase, they results in the formation of oxaloacetate plus ADP plus IP. So these are the important classes of enzymes, which this classification was made by IUB in 1961. So important classes are 6. One is oxido reductases, which helps in oxidation and reduction reactions. Second is transfer of one functional group from one substrate to the another, and hydrolases or the enzymes which helps in catalyzing hydrolysis of substrate molecules, or they helps in hydrolysis reactions. And the next is lyases, and these lyases especially they helps in non-hydrolytic removal of functional group from one substrate to, and cleavage of covalent bonds. And next is isomerases. They helps in converting one isomer to the another isomer, and lyases especially helps in the formation of new bonds. These are the important groups or classes in this enzymes. The next mechanism of enzyme action. And this mechanism of enzyme action, what we are going to discuss is how the enzymes they works with the substrate molecules. all the enzymes in the same pattern, or is there any difference? Yes, there are two mechanisms. One is lock and key theory. The other one is induced fit theory. Let us discuss about the lock and key theory. What is lock and key? We know that lock, for every lock, there is a special key. Then only that by putting this key, that lock opens. Here also the same principle. Here, lock is enzyme and key is substrate molecule. So, for each and every enzyme, there is a special substrate molecule. For each and every substrate molecule, there is a special enzyme. And this proposal was given by Fisher. Fisher scientist, he proposed this lock and key theory. And according to this, the structure of the enzyme acts as a lock and substrate fits into the this lock as a key. That means that this acts as a key and enzyme acts as a lock. To understand this, if we observe the structure of enzyme structure, you can understand this lock and key theory very easily. This is the shape of the enzyme. Just imagine, and that is in this way by having this active sites. And in the same shape, in the same similar structure, substrate molecule is showing the structure, isn't it? So in this active sites or in this depressions, these depressions are called as active sites. So in this depressions or in this active sites, this enzyme comes under, this substrate molecule that comes and enter into this depressions or active sites. So this depressions or the active sites into which substrate molecules is going to fit in it. Okay, so lock and key. Here, enzyme is acting the role of lock and the substrate molecule is playing the role of key. So this indicates yes. Okay. This right. So according to the enzyme shape, substrate molecules are constructed. Right. And what about the reaction between these two? That is explained in this slide. See here, substrate molecules, they are in this shape, triangle shape. There are two substrate molecules. Okay. Here we are taking two substrate molecules. We are taking two substrate molecules and which are fit into the active sites of the enzyme. See here, this is the enzyme and these two are the substrate molecules. And these two substrate molecules, they have to fit in the active sites of the enzyme. And basically what happens here means active sites of enzyme and substrate molecules both makes a bond. That means the substrate molecules, they makes a bond with the active sites of the enzyme. And this is specific. That means each substrate have its own specific enzyme. All the enzymes cannot work on the same substrate, or all the substrates do not have the same type of enzymes. Each substrate have its own specific enzyme. So according to that, these substrate molecules, they reaches their respective enzyme active sites, and there they makes the bond with the active sites and become specific. That means a specific substrate can make a bond with the specific enzyme. there is a specificity between the enzyme and substrate molecules. So after recognizing this specificity, this enzyme active site that receives the substrate molecules, and this substrate molecule, they comes and fit in the active sites, and they develops bonds, they will develop bond with the active site and becomes a complex, which is called as, first of all, initially enzyme is separate and substrate molecules are separated. During the reaction, according to the specificity, this substrate molecules comes and fit in the active sites, and they develops a bond with the active sites, and by that they becomes a complex, which is called as enzyme substrate complex. Now this enzyme substrate complex, that see here, as soon as both the substrate molecules comes closure and bond formation takes place between S1 and S2, there is a temporary bond, and that complex is called as enzyme substrate complex, and this is temporary. This substrate complex, enzyme substrate complex state is temporary, and furtherly it dissociates into product, and this product, here they are separate, but by the association of this enzyme active sites, they both substrate molecules, they comes closure and makes a bond with one another, and furtherly it becomes as a product. So here, from this enzyme substrate complex, further reactions takes place, and at the end of these reactions, enzyme comes out as it is without undergoing any change, and this substrate molecules, they comes closure and makes bond with one another and results in the formation of product, end product, and they comes out. Here it is a product. So this complex dissociates easily and gives a product and enzyme without any change. That means here substrate molecules 1 and 2, they have attached with each other by bond formation, and furtherly they are called as product, end product. So this is about the lock and key theory. That means by putting these substrate molecules in this lock, like sub enzyme, this substrate molecules, they reacts with one another and helps in giving a new end product, and this lock or enzyme that comes out as it is without undergoing any change. So this is about a lock and key theory. Once again, in this mechanism of enzyme action, there are two theories. First theory is a lock and key theory, which was proposed by Emil Fischer. According to him, each and every enzyme has its own shape, and according to that enzyme, substrate molecules are framed. And here, enzyme is compared with the lock, and substrate molecules are compared with the keys. So here, for example, this is one enzyme. This enzyme, it consists so many depressions, which are called as active sites, and this active sites helps in receiving the substrate molecules with their respective or with their similar shapes of substrate molecules are received by this active sites, and after receiving these substrate molecules, they comes and fit in the active sites of the enzyme, and they makes bond by coming closure with one another. The substrate molecules, they makes bond, they forms or develops a bond between them, and finally they comes out in the form of end product. So here when the substrate molecules are associating with this enzyme, they develops a temporary product, which is called as enzyme substrate complex. And this enzyme substrate complex is temporary. Furtherly, that undergoes changes and gives enzyme acidity and end products. That means by the association of a substrate 1 molecule with substrate 2 molecule, they they forms a new bonds and becomes a end product. Here, here, what is happening? This substrate molecules, they are separate, but by the association of enzyme, they come in contact with one another, or by coming closer, they develops bond between them, and they comes closer, and they connects with one another and makes a temporary bond, which is called as enzyme substrate complex. And this complex dissociates. It's a temporary state, and that dissociates into enzyme and the end product. So here, what we are saying, enzyme is acting as a lock, and substrate is acting as a key. So according to the lock, key is framed. So based on that principle only, to open the lock, this key should be in the shape of a lock only. That's why then only there is a chance of fitting with the lock. Then this complex, it is temporary. After removing this key, again lock come out in the same shape. So based on this principle only, this theory was proposed by Fisher. So how the action will be means, this enzyme have active sites. By having this active sites, it receives the substrate molecules which are similar in shape of this lock active sites, and by joining with the active sites, this substrate molecules they fit into the active sites, and by this what happens, the substrate molecules, they comes closer with one another, and they forms bond, temporary bonds with one another, and by this overall a complex is developed, which is called as enzyme substrate complex. And this complex, it is temporary state, and furtherly it dissociates into two products. One is enzyme, which do not undergo any changes during the reaction, and end product. Initially, this substrate molecules, they are separate but by the association of this enzyme, they undergoes changes and becomes together. That means these two substrate molecules, they comes closer with one another and makes a new bond formation and results in the formation of a new end product. So this is about the lock and the key theory, which was proposed by Fisher. And the second theory is induced fit theory, and that will be explained in the next class. Thank you.